Okay, so on to another topic that is very important. This is one of those like merging fields in biology that has great potential to it. The application of the DNA into a particular task. Can you use it for solving a particular problem? So really what we want to do here is recombine the DNA. Notice it's talking about two species, and it means just that. So perhaps you have a bacterium, and then you have a piece of DNA for something else, another species. Put it into the bacterium, glue it together, put it back into the bacterium, and then get that plasmid in the bacterium to do something specific for you. Maybe insulin. So maybe you're a diabetic and say pre-1970s. At that point, maybe you only had pig insulin or cow insulin available to you. And while a mammal's a mammal and you're a mammal, now you have the ability to take that bacterial plasmid, put the human insulin gene in, glue it back together, put it back into the bacterium, and get it to make pure human insulin. You are then combining the DNA. That's genetic engineering. And so it to say it revolutionized is an understatement. This is hugely impressive to be able to solve a problem in a novel way that might be more efficient than what you're used to using, what you had before. So it's really quite impressive. Also including microarrays, see how DNA expresses in different environments. Actually, it's this topic which led to my becoming a professor at HEC. I'll explain more about that later on. So we're going to clone, and we mean just that. You're going to, again, like the insulin, clone the DNA again and again and again, and then get that DNA to do something for you, in this case, make insulin. So that plasmid, that's a word I already used. It's a small piece of DNA that's offset to the larger bacterial chromosome. And so you could take that plasmid out, crack it open, chemically crack it open, put in the gene you want, glue it back together, put it back into the bacterium, and again, get it to make what you want over and over and over and over again. So it is recombinant plasmid. It's a mix of two different organisms in that bacterium now, getting it with the foreign DNA, maybe it's human DNA, to do a particular task for you. So here you go. There's that bacterial chromosome. There's the plasmid. You crack it open. You put in the gene you want. Again, maybe the gene for insulin. You take that, now glue it together. Use ligase to glue it back together. We've seen that before. Put it back into the bacterium, and then just get the bacterium to divide. And it doesn't know any different. It's just a piece of DNA. So it makes insulin, and therefore you're able to use it for something. Or you use it for plants. Maybe you make, here they're saying resistant to something. Maybe it's resistant to pests or to salt or something like that, or oil spills, although there's other techniques you can use too, or solving human health issues, whatever it may be. It's recombinant in that two different organisms start really coming together, in this case, human gene and a bacterium, to try to solve a problem. So that idea of cracking open the plasmid is with this restriction enzyme. It cuts at very specific points. And then when it cuts, it breaks open the bacteria's plasmid, but it doesn't do it like evenly. It's not a clean cut. You have these like overhanging ends. And we call those overhanging ends sticky ends. Then you put in the piece of DNA you want, glue everything back together. There's the DNA ligase I just made reference to, and then you put it back into the bacterium. So there you go. It's just an enzyme that looks for a very specific site. It cuts at that site. You can see the little overhangs I was just talking about. Then you take the DNA you want, you put it in, glue it back together, and then you put that back into the organism, back into the bacterium. So we've been doing this for a while now, which means we've been able to create effectively libraries. There's two main ones we talk about. There's a genomic library and then a cDNA library. It's with the genomic library, it's like a collection of smaller pieces of the entire genome, and then you have like one piece in each bacterium, and then you would like add all the bacterium together to get all the genome built back up. It's just a way to like make it more manageable, the small pieces, looking at the entire puzzle. You can use the bacterial artificial chromosome. This is a large plasmid. Notice it's trimmed down. Or you can use, again, the complementary DNA. You actually use reverse transcriptase, so mRNA to the DNA. And then from that, you would put it back in the bacterium and then build your library out. Now, it sounds direct enough, but it's actually a little bit complex. So I thought I would show it to you. Turn on the sound so you can hear this. Okay, so as always, when I play video, I can't say anything, but hopefully you can hear the video well.
Hello, and welcome to another help session on recombinant DNA. Today, we're going to discuss genomic libraries and cDNA. So what is a genomic library? Well, with most organisms, the genome is really too large to deal with, too large to work with as designing. So oftentimes it's more useful to cut it down into smaller pieces so you can run it down. How do we make a genomic vector? Well, we start off with our cell interest. And the first thing that we're going to do is we're going to isolate all the DNA from the cell. Once we have the DNA, we're going to use a specific enzyme to cut it into smaller sections. Now we have to be very careful when we choose which recipient enzyme we must use. We want to use an enzyme that has enough restriction sites in the genome so that we'll get small enough pieces cut in the vector, but not so small that all of the genes get cut off. Usually what we'll use as a vector is a plasmid. Plasmid is a very standard and easy way to get foreign DNA into bacteria. So we're going to cut up our DNA and the plasmid with a specific enzyme. Sometimes you might even want to make two different libraries using two different restriction enzymes so that we'll have the cuts in different places. Once we have our cut up DNA and our plasmids, now we need to ligate them together using DNA ligase. Once the cDNA is pasted into the plasmid, now we're ready to actually transform them into bacteria. Very easy to transfer plasmids into bacteria. There are multiple different ways to do it. One common one is to use heat shock, whereby you heat the bacteria up and then cool it down rapidly, which causes new holes to open up that allows the plasmid to open up. What we end up with are thousands of different bacterial cells, all with different sections of the DNA. So combine all these different sections from the genome, the original genome we have, but they're much smaller sections than much easier to work with. So this combination of all these different cells forms the genomic library. So that's one type of library. Another type of library is a cDNA library. So the way that the cDNA library is different from the genomic library is that while the genomic library is the entire genome of the cell, the cDNA library just looks at what genes are being expressed in the cell. So let's go over how we would make a cDNA library. What we're going to start off with is we're going to start off by isolating the mRNA. So the mRNA, clearly, the RNA that's being expressed in the cell. This is what's also known as proteins are going to be produced by the cell. We're then going to use reverse transcription to create the DNA version of the RNA, what we're going to present as the cDNA. Once we have the cDNA, we then need to add restriction sites to the end step. Remember before, we were just able to use the innate restriction sites in the genome. So the cDNA library, we really want to keep the cDNA intact. We don't want to cut it off. So we just want to add restriction sites to the end of the cDNA. Then we can treat the cDNA and the plasmid with restriction enzymes. Again, once they've been cut up, we can use DNA ligase, combine the cut up plasmid and the cut up cDNA to the complete plasmid plus cDNA. And finally, once again, we can transform our bacteria. And then a combination of all these cells, all with different types of cDNA, form a cDNA library. The cDNA libraries are very useful if you want to compare the difference in protein expression between these different cells. Thank you very much. This is another help session for programming DNA.
So I just wanted to note, these MIT series are free and just, in my nerdy opinion, fantastic. There's just so many different ones out there, not just in biology, geology and astrodynamics, and you name it. It's just brilliant what's out there for you to see. And I can't remember coming across an MIT one that I was like, huh, on the science. I've said it before about YouTube. YouTube's YouTube. There's a lot of things on YouTube that are great and some things that are not so great. But these MIT ones I think are quite nicely done. So there you go. There's your libraries. Now, another way you can do it is maybe you're after a certain sequence. So you tag, you say you want this. So you make this tag, and this tag, notice GCAT, GC, it looks for that, and it kind of like hybridizes with it. And therefore kind of like says, hey, it's here, that kind of idea. So there's a lot of different ways in which you can clone, a lot of different ways in which you can make something. We're going to look at several different processes. This chapter has some, just be aware, like what southern blotting is and northern blotting, but then also has some applications that we'll get to as well. So one of the problems is, what do you do if you're working with bacteria, but you want to apply that to eukaryote? There's obviously similarities, cells are cells, but there's also some significant differences. So that's what this one is. How do you differentiate that and work with bacterial versus eukaryotic? You would use maybe an expression vector. You can see exactly what it is here. Or you might try to find something that's a bit of an in-between. They were noting yeast might be an in-between that can possibly work. If that doesn't work, you might try, again, something else. Here's mammalian cells or insect cells. You look for a way to find like a bridge to get to where you want to go. Now that gentleman in the video was mentioning heat shock and opening up. This is electric shock and opening up. And you have these little holes that open up, and then you can help get that DNA into it. Even microscopically thin needles can be used. Just to show you real quick, this is not, I mean, it is biotech, but it's not biotech that we're talking about. This is actually a sperm. And to show how small these needles are, this is a human egg. So this is a version of like in vitro fertilization. But this is a micro needle. And you can see it's incredibly small, a single sperm right there. There's a head of a sperm. They position this, and then they actually take that needle, puncture the egg in, and then cause fertilization to take place. So they aren't joking when they said that these are really, really small needles, micro needles. So just to give you an example, I mean, this is still a biotechnology issue. So gotta position the egg properly, of course. And here we go. Almost. Like I said, you gotta get that really well positioned. Right. And here we go then. So you get the sperm. That's a single sperm, mind you, right there to the end, and then puncture in and deposit the sperm in. It's absolutely phenomenal what you can do with this biotech. You would think it destroys the egg, but these techniques actually work. So that plasma membrane is tough, and there we go. Finally, in the plasma membrane, they pull back a little bit of the cytosol, and then they move the egg, the sperm in. Excuse me. So you can see they're drawing back a little bit of the cytosol just to make sure they have good penetration into the egg. There you go. And then you still see the head right there. Et voila, you have a fertilized egg. Isn't that incredible? And there you go. So this idea of microscopically thin needles was not meant to be a joke. It's really quite incredible what we've created. Here's another one, and I hope you get to do it in the labs at HCC. Polymerase chain reaction. You can combine this with gel electrophoresis. This is where you have just like a little bit of DNA and you need to amplify it. So maybe you have like a little bit of blood or a little bit of saliva, a little bit of semen or something at a crime scene. But it's not enough to do genetic work on. So you put it into a machine, a thermocline, heats it up. This thing called Taq polymerase, which is, there it is, right there. Anneals to it, copies it, cools it down, and now you go from one copy to two, and then you do it again and again and again, and you can go from one copy to like a million within a short amount of time. So here's a sequence you want. You denature it, you copy it, and you end up with two, and then you just do it again and again and again and again. So I wanted to show that to you as well. You don't need any sound with this. Actually, there's some good sound here, so I think I'll include it. Okay, there you go.
Polymerase chain reaction, or PCR, uses repeated cycles of heating and cooling to make many copies of a specific region of DNA. First, the temperature is raised to near boiling, causing the double-stranded DNA to separate or denature into single strands. When the temperature is decreased, short DNA sequences known as primers bind or anneal to complementary matches on the target DNA sequence. The primers bracket the target sequence to be copied. At a slightly higher temperature, The enzyme Taq polymerase, shown here in blue, binds to the primed sequences and adds nucleotides to extend the second strand. This completes the first cycle. In subsequent cycles, the process of denaturing, annealing, and extending are repeated to make additional DNA copies. After 3 cycles, the target sequence defined by the primers begins to accumulate. After 30 cycles, as many as 1 billion copies of the target sequence are produced from a single starting molecule.
And there you go, 1 billion copies after 30 rounds of this, just absolutely incredible. And so it allows you to do all sorts of things, compare genes between people. So for example, you might be doing like, I don't know, Ancestry.com or 23andMe or Living DNA, and you're trying to differentiate very close groups, like in my grouping, the English, Irish, the Welsh, the Scottish, the Manx, very close groups, especially the English, the Irish, the Scottish. How do you differentiate them? Really hard to do. So you might be trying to do that, very small differences in that regard, or find out if something exists in an organism or what it does. I mean, there's a lot of technique you can do here. Like I said, you can maybe copy the DNA and then run it on a gel. Gel electrophoresis, it looks like Jello actually, but it's actually a matrix. And so you take the DNA you want, you use these enzymes to cut it up. It looks for very specific points to cut. When it does cut, you get different size fragments. You apply electricity to it. Now, DNA is charged, and so it's going to run. Let me show you here. See, it's charged. It's going to run from those phosphate groups being negative to the positive end. So it's going to run to the positive end. Now, these fragments that cut is very specific, these restriction enzymes when they cut. So this is so specific, you can actually use this, from my understanding, in criminal cases. There should be nobody on the planet has the exact same DNA as you except an identical twin. And even then, can we look for any kind of environmental changes, epigenetics, that kind of idea. But so let's say, for example, my sister and I, she is the closest genetic person to me on the planet. So these enzymes, they would cut differently from her than me, even though we are the closest person to each other there are on the planet. She is my natural sister. So I would have slightly different cuts, which means the banding pattern that I would get, let's say that's mine, would be different than hers, and clearly able to tell our DNA apart from each other. So it's really quite amazing. Now, again, you cut it up, you have to use these micropipettes to load properly into these wells, these like little depressions in the gel. You apply electricity, again, the DNA is slightly negatively charged, so it's going to run to the positive end. As it runs to the anode, the smaller fragments move faster, the larger fragments move slower, and you get what's called a DNA fingerprint. Now, this being said, it's quite a technique. Last time I was teaching labs, I was showing students how to do it. Very hard to get good results the first time through. I tell my students not to worry about that, to worry more about the techniques, to feel what it's like to use micropipettes, to load a well, to run the DNA, that kind of idea. They get great results, outstanding, but just being exposed to how to do the techniques is invaluable, in my opinion. So I thought I would show you that as well. Change my sound real quick.
Welcome to the Kirkwood Community College Biotechnology Informational Videos. Today we'll be doing gel electrophoresis. The general electrophoresis equipment includes a power supply, gel mold and form, and gel box. Here we have the gel mold, form, and 2 combs. Carefully take the mold and place it into the form and place your 2 combs in. Choose the combs according to how many samples you have. I have a total of 10 samples, so I will have 2 8-well combs. For these particular samples, I prepared a 1% aluminose with amphibian bromide and 1x TAE buffer. When working with chemicals like amphibian bromide, ensure you're wearing proper personal safety equipment. As you pour the gel, ensure you do not form any bubbles. Also, when you are done, check the bottom of the combs just to ensure you also do not have bubbles there. When your gel turns opaque, it will be ready to use, as you can see here. Next, we must prepare our samples for loading onto the gel. I have a total of 25 microliter reactions. Therefore, I will be using 2.5 microliters of a 10x loading dye. This loading dye will contain 2 dyes so I can monitor electrophoresis and also glycerol to help weigh down the samples into the wells as I load them. To prevent contamination, ensure you use new tips between each sample. Repeat these steps until all your samples are prepared for loading. Now you're ready to load your gel into the gel box. Carefully remove your gel from its form and place into the gel box. Keep in mind your DNA will migrate to the positive pole due to the negative charge in the phosphates in the DNA. Today I'll be using 1xDAE buffer. Carefully pour the buffer into the gel box, filling both chambers. All you need is a thin layer covering the top of the gel. When this is done, carefully and slowly remove the cones from the gel as do not tear the gel accidentally. Improper removal of the cones may cause a hole in the bottom of the well and cause leakage later as you load your sample. It then may be necessary to top up the gel with more buffer to ensure that the wells are completely covered. Now we can load our gel. I will be using a 2-log DNA molecular weight monitor. This will allow me to later estimate the size of my tank and my gel. When you are loading, in this case, I will load my molecular weight marker twice, one in each row. Now it's time to load your samples. As you load your samples, be very careful not to puncture the bottom of the well with your tip. The agarose is very thin, and if that is done, your sample will leak at the bottom. Also ensure that in your lab notebook you note what samples will go in what wells to avoid further confusion. Now we can start your electrophoresis. Carefully put on the lid and making sure your electrodes are hooked up properly. Remember, your DNA will move towards a positive pole. Turn on the power supply. In this case, set the constant mode, and I'll be running this gel at 130 volts. This may vary based on the type of gel, percentage of gel, and how large the gel is. Always a good idea to make sure electrolysis is taking place, so look into the gel box and make sure bubbles are forming. And here we have electrophoresis as a time lapse. You can see the dyes migrate. I like to do it until the dyes divide the gel into thirds. When electrophoresis is done, turn off the power supply, carefully remove the lid, remove the gel from the gel box, pouring out the excess buffer, and place onto a paper towel to absorb excess liquid. Now it's time to document the results. I will do this at a later video.
And there you go. And then you would put it under UV lamp and you can see these nice DNA fingerprints. And this is specific. Again, I've read about court cases where they bring this in as long as the techniques are done properly. So you might be looking for very small changes in the DNA, maybe a single nucleotide polymorphism, like a single base change, or changes in these restriction sites. This will pop up a few times in the lecture, restriction fragment link polymorphisms. So maybe again, where it cuts is a little bit different from one person to another, that kind of idea. So notice here, this is somebody who doesn't have sickle cell anemia, and it cuts a little bit differently than somebody who does. And you can see when you run the gel, you get a different DNA fingerprint. Now, there's actually several kinds of blotting techniques out there. There's Southern blot, Western blot, Northern blot. Your author brings up two. I'll show you a little video on it in a moment. Notice here you're using gel electrophoresis with DNA fingerprints. So it's a DNA fragment that's running. Just to show you, that's a little bit down here. I'm sorry about it. I'll come back. Don't worry. Northern blot. Notice it's gel electrophoresis, but with mRNA that you've been using. So a little bit different idea. And then you're going to get sort of a different kind of banding pattern. Let me go a bit further here, and then I'll get back to the southern and I'll show you a video on that. Here's another one. This brings back tons of memory. Here you have chopped up DNA and you're fluorescently tagging the ends of it. And so what is that fluorescently tagged in? Is it an adenine, a thymine, a cytosine, a guanine, that kind of idea? And you would have a smaller fragment, a little bit bigger one, a little bigger one, a bigger one, a bigger one, until you've built up the entire chain. And since you're only looking at the very ends, you're building up the A, T, C's, and G's until you have the entire sequence. They're known as the deoxy chain termination method. So notice this is DD and TP, but you're really looking at what is it? Is it DDC, so a cytosine at the end? And then what's next in the list? Well, it's a G, so DDG, and then next in the sequence, DDA, then DDA, then DDG, and you're building up the sequence. And then a laser reads it, and it sort of shows you what the DNA sequence is. And why this brings back memories, I had a good friend, several good friends actually, during my master's who was at USF. And so we would go and have like lunch together. And I was in the ecology side and they were in the molecular genetics side, two very different biosides. But you go by their door and they would have another one of these beautiful graphs, like here's what we sequenced today. It's some genome that they were working on. So it's pretty cool to be able to see this and that they were using not just technology, but lasers to build up the DNA sequence. Pretty cool. So again, you're looking to see how the DNA is expressing. You have all these different techniques. You have the southern blotting. You have the fluorescent tagging here. And then, of course, you have the northern blotting. And as I was saying with the northern blotting, this one, instead of using DNA with the southern blotting, here you're using mRNA. So a little bit different idea. And then even this one, reverse transcriptase polymerase chain reaction, RT-PCR. Notice it's more sensitive to the northern blot, and it sort of gives you, again, an idea of what the DNA is doing. But in this case, you're using complementary DNA. So there's a lot of different techniques here. So I wanted to give you a little bit of a rundown on that. All right, let me change my sound again.
Welcome back. Today we'll be talking about the blotting test. And I'll try to cover everything that you need to know for your step 1 exam. Before we jump into the mechanism, you'll have to know that we use the blotting test to detect the presence of certain segments of DNA, or detect the presence of certain gene expression, or the presence of a specific protein. And this is how this is done. So we'll start with a sample of DNA that may or may not contain the certain segment of DNA that we're looking for. We'll use a cleaving enzyme to take off one strand of the DNA. And we'll add a radio-labeled primer that is specific to that DNA that we want. For example, if we're looking for HIV DNA, we'll add labeled primers to that HIV DNA. If that sample of DNA did in fact contain some HIV DNA, this primer that is specific for that HIV DNA will link to this segment. And then we will be able to detect it. Now, the process of detecting a specific protein is a little bit different. We start off with a sample that may or may not have this certain protein that we want. Say, for example, a sample of cells that may or may not have HIV proteins. We'll add in antibodies to HIV proteins. And if the sample did in fact have HIV proteins, these antibodies will link to this protein and we will be able to see them. Now we use the southern blot to detect DNA like we just described. And we use the northern blot to detect gene expression by looking at mRNA. So if we have mRNA of a certain gene, that means that this gene is expressed. And most famously, we use western blot to detect specific proteins, most importantly HIV antibodies. And we can use the southwestern blot. From south we get DNA, and from western we get protein. So southwestern is DNA-bound proteins. This includes all the proteins that are attached to our DNA. So I think transcription factors, histones, etc. And here's a fantastic mnemonic to remember the blot tests and their functions. Whenever somebody's talking about blotting tests, think about snowdrop. Alright guys, that's everything I've got. Hopefully I made this easier for you. Thanks a ton for watching. And
so in this case, they went through a little bit more than what we did. They just we just had southern and northern blotting, but it's okay. It gives you an idea. There's a lot of ways to look for it. Another way is to try to express in situ, means in the organism. So what genes are there, what genes are expressing, and therefore get an idea maybe what the mRNA is up to. So again, there's a lot of techniques here that you can do. Microarray, yes, so I mentioned that this is one that actually is, in a way, the reason why I'm in front of you. In 2004, I was actually offered a job working with a woman who I thought had the most fantastic research. She used C. elegans, which is a kind of nematode like a worm, shot up into space on a space shuttle, let it float around in space doing wormy things for a few weeks, brought it down, and then looked to see how was the DNA changing from a gravity field of Earth, negative 9.8 meters per second squared, to a microgravity field of space. And then you're looking to see, again, just what is the DNA up to? So this idea is, again, the worm. It would be a tissue sample. Yes, I'm very sorry. The worm might have to be killed. Again, unfortunately, in science, it's still kind of the way it works. It's not something I liked, and it would have been a part of the research I would have been a little bit unhappy about. But I mean, it's no different than like going fishing. You're just using a worm in this case, but it is still a living organism, I agree. Then you make cDNA by the reverse transcriptase, you fluorescently tag it, so you label it, then you put it into what looks like, it's called a stamp, but it's all these like little wells that are available, each having a little bit different DNA. And then you look to see where the cDNA hybridizes. So you put into the cDNA you made, and then you wash it off, and you look to see, is there any spot that now fluoresces, and that would be its complement. So to give you an idea of that, I have a video for you as well.
A microarray is a small silica or glass slide, which is the size of a postage stamp, that contains thousands or even hundreds of thousands of spots. Each spot contains a synthetic piece of DNA with a known sequence. A microarray consisting of partial sequences from a number of specific genes is created by placing a series of known single-stranded DNA sequences in an array on a microscope slide or a silicon chip. The sequences are placed in a specific pattern so that each position on the slide corresponds to a particular known DNA sequence. The portion of a microarray shown here only contains 6 spots, which are labeled A through F. These spots may correspond to DNA sequences found in 6 different genes. The expression of genes depends on the cell type and the environmental conditions. To study gene expression, messenger RNA or mRNA can be prepared from a particular type of cell, such as a muscle cell, or from cells that have been subjected to an environmental agent, such as a hormone. This mRNA that is expressed from such cells is used to make complementary DNA or cDNA. cDNA is made in a test tube by an enzyme called reverse transcriptase. This enzyme makes a DNA strand complementary to RNA molecules used as a template. Special nucleotides that have a fluorescent probe attached to them are used to make this cDNA. This probe will make the cDNA molecules fluoresce green when exposed to ultraviolet light. Once the fluorescent cDNA has been prepared, it is allowed to hybridize with the DNA samples spotted in the microarray. The cDNAs complementary to the type A mRNAs will hybridize with the DNA spot in the array that contains DNA complementary to the type A cDNA. Likewise, the type D cDNA will only bind to the DNA in the spot labeled D, while the type F cDNA will only bind to the spot labeled F. Since the cells did not express any mRNAs that correspond to the DNAs in the spots labeled B, C, and E, no cDNA is made that will bind to these spots. After hybridization, any unbound cDNAs are washed away, and the slide is examined under a fluorescence microscope, where the spots that contain fluorescent cDNAs can be identified. In an actual microarray analysis, a typical cell would express hundreds or thousands of different mRNAs, so that hundreds or thousands of different spots would be identified. Therefore, microarray analysis is a powerful method to study the expression of many genes simultaneously from a particular type of cell or from cells that are exposed to a specific environmental condition.
So just to finish out the thought, the reason why this has connection to me to HCC was, even though I accepted the job and I was on my way out to do the job, it was a government job and the job got canceled before I even walked in the door. So I never even got a chance to try it, and I scrambled and ended up at HEC. But I just thought it would have been fascinating to be able to do that. But oh well, didn't get to work out. So another one in the organism, so in vitro, excuse me, in like test tube materials, mutagenesis. So you're causing a mutation to be introduced, and then you want to see how it affects the phenotype. Yep, there it is, the phenotype. Kind of like what we saw back in like the X-raying of gene material to see how that affected enzymes. Remember the one gene, one enzyme hypothesis, which got later rewritten to the one gene, one polypeptide hypothesis, that kind of idea. And you can even introduce the RNAi, as we talked about last chapter, to block the DNA directly. There's a lot of ways you can just see, well, what is being affected by the DNA, and therefore you get an idea, the proteins, and maybe then the phenotype. So here's a phrase I used a moment ago, this little acronym, single nucleotide polymorphisms, which is just a single base change that allows for something to occur. And I referenced it in regards to, like Ancestry.com and 23andMe. And I was saying that in my group, the Irish, English, or Welsh, or Scottish, they're so similar. There's so much mixing of DNA over the centuries that it's really difficult to pull it apart. I do caution people, if you ever do like Ancestry or 23andMe or family tree DNA, whatever it is, it's not just the genetics. You can't just say, oh, my DNA says. And bear in mind, even small percents mean a lot, especially when there's a lot of mixing going on. But you got to combine that with oral history, and what did your family say, with historical documentation. So again, this is one piece of the puzzle, but it allows you to look for something, to look for something specific. And notice here it says disease causing. So, for example, this would have been the wild type, and here would have been the disease. Well, let me give you an example of that. So in my group, Scottish and Irish, one disease often comes up, and that's celiac. And I've been tested for it. So far, I'm fine, which means I can still handle things like wheat, rye, that kind of thing. I don't eat it very much, even though I do like breads. But notice down here what it says. A study of over 27,000 subjects identified this single nucleotide polymorphism, what do you know, as a primary genetic factor for celiac disease. People carrying the TLL had over 6 times higher chance of being diagnosed with celiac disease. So it is a single nucleotide polymorphism. What do you know? Now, I haven't been tested specifically for this variant. I don't know why I haven't, but I could have that done to see if I'm at risk of it. If I remember correctly, one of my genetic tests says I'm at an elevated risk of celiacs, but it doesn't mean I'm actually going to get celiacs. But I have to be careful of it. So, I do my yearly physicals. I see what's feeling right for me, and I don't eat a lot of bread anymore, even though I really like bread. It's just something I don't imbibe as much anymore. But it's something you can use an S&P for. So with the cloning of organisms, again, you're going towards application. It could be anything from agriculture to foods to just disease issues, maybe making a new drug. There's a lot of things you can do with this. Notice here organisms, so you want them to be identical to the parent. Well, that gets right into cloning. Cloning and stem cells, two very different ideas. Make sure you separate them. Even this, totipotent versus pluripotent. Totipotent means from that cell, you can actually make everything, make the entire organism. Where pluripotent means you can make a lot of cell types from that, but maybe not the entire organism. Here they're bringing up plants. So you can see from what looks like a carrot to me, they scraped off some of the cells, some of the embryonic cells, or just some of these cells that are totipotent, I should say. And they're able to clone the entire organism back again. So they're able to effectively make a clone of the adult plant. I mean, it's really quite amazing what you can do with this. Now, nobody would care if you're cloning carrots. Heck, bananas. You're eating effectively a cloned banana when you go and get the Cavendish, a banana like at Publix or Sweet Bay or Sam's Club or wherever it is. I think I mentioned that before because of the worldwide fungal outbreak back in the 1950s. So again, nobody really cares about it there. But when it comes to the Animalia, certainly the Mammalia, people tend to care. So when you get more towards that, okay, so I'll leave the bioethics to you, but this is not easy to do. Technically speaking, the process is direct. You have a cell, you take the nucleus out you don't want, you put the nucleus in you do want, and then you see if you can rear it out. So that's what they're trying here. They tried it with frogs, and notice same thing. Here's the cell, you took the nucleus out you don't want, and you put the nucleus in you do want. Now notice on this side, they put in a nucleus from a cell that wasn't differentiated yet. It was still more or less like totipotent, so it was able to clone out. Here, they put in a differentiated cell, and it didn't work out. So it took us time to figure this out. Now, a tadpole, most people wouldn't care much about a tadpole, amphibia, although, it's still an animal like anything else, but this. So this is Dolly, Again, Dolly was back in 97. This was a mammal, obviously, same kind of idea. So they cultured the cells, they took out the nucleus that they want, took the nucleus they want, put it back into the cell that now has an anucleated cell. So now it has the nucleus you want in the cell, you put it back into the donor, and you got the identical organism back out again. So again, she was identical to what you wanted. But this was back in 1997. She wasn't very healthy. She did lead to premature death. But we've gotten much, much better. I've read about cloning techniques now where, you got to spare $25,000 lying around. You can clone, your cat, or I think dogs are 50,000, horses were like 100,000. It's come a long ways since 1997. It doesn't mean we get it right on the first shot. Kind of keep that in mind. And even then, you clone like your favorite cat, but you said this is the best cat ever for me. Remember, we're a makeup of two things, our genetics and our environment. Does anything say that you're going to get the exact same cat out again? Yes, you get genetically the same cat out again, but are they getting the same life experiences? Where cat one might have been the sweetest thing around, who knows? Cat two might be like hell on wheels. So again, kind of be careful with this, and I'll leave the bioethics to you. What I'm getting at is this: carbon copying. Yes, it has happened. Yes, we can do it. Is it okay? Well, there is some application here. And I'll give you an example.
Some people think that cloned cats are creepy, but what about a cloned cat that also glows in the dark?
It's an incredible story. Not everybody's happy to hear this, though. The scientists in South Korea cloned a pair of kitties who light up with a fluorescent glow when exposed to ultraviolet light. Glow kitties.
All right, now the question is, what was all this for? Apparently there's a good reason for it. Christy Lustow has the story.
At first glance, these cats look like any normal cat, but there are two big things which make them very different. One, the cats are clones. Two, they glow in the dark. You heard right. When they're put underneath ultraviolet light, they glow a dull red. And it's no accident. Scientists manipulated the donor cat's genetic code, then passed those changes onto the clones.
It's meaningful as we introduce outside genes to transgenic clone cats for the first time, and as they have the red fluorescent protein gene in their organs, they give off a red color.
Here's how they did it. Scientists took skin cells from a Turkish Angora female cat and used a virus to insert the genetic instructions for making red fluorescent protein. Then they put the gene-altered nuclei into eggs for cloning. After that, they implanted the eggs back into the donor cat, which effectively became the surrogate mother. Now, these glow-in-the-dark cats shine a light on what may be possible down the road. Scientists say if you can pass along coding for fluorescent markers through cloning, you could eventually pass along more complex genetic coding. That means cats could help develop treatments for diseases, for animals, and for humans. After all, researchers say there are some 250 diseases which affect cats and humans. In theory, leaving a warm, healthy, fuzzy glow, many more than just their 9 lives. Christy Lustow, CNN, Hong Kong.
So there you go. Now, one thing I would note, animals versus humans, that's not good science. A human is an animal as well. So a mammal's a mammal. Yes, this is in a different order, the order Carnivora, we're in the order Primata, but still there's connection there. So interesting. And again, I'll leave the bioethics to you. So again, they do have problems. These epigenetic changes are out there. This is change to the DNA that doesn't, its effect on the DNA that doesn't include actual rewriting the DNA. So notice here they're mentioning acylation. We talked about acylation versus methylation. So here you're loosening the DNA. You didn't change the genetic sequence, but you loosened it, which might speed up the transcription, or you methylate it, which will tighten it, which might slow down the transcriptional process. I should say transcriptional process for both. So this increases transcription and this slows down transcription, but you didn't rewrite the DNA. So can you do it accurately? Can you do it with efficiency? Again, that might be a little bit more challenging. Are you always getting a proper kitty every time, sort of idea? So that's cloning. And again, cloning has to be differentiated from stem cells. People love to lump the two together. Please don't do that. stem cells or undifferentiated cells that become a lot of different cells. Now, that doesn't mean they're always totipotent. And there are two kinds here. There are embryonic stem cells, which automatically brings up moralistic issues. And there are adult stem cells too, right there. They're not as useful, perhaps, as adult stem cell, although I read about a Japanese research team years ago that were able to get adult stem cells to function like embryonic stem cells, which, who knows, maybe they'll take away some of the bioethic issue that's out there. But the idea being the embryonics, I mean, look at how many different things you can use embryonic stem cells for. But with the adult stem cells, maybe not quite as much. But the idea is to create cells. So one thing they might do is collect this from placental material from you when you're born, and therefore put it on file in case you need your own embryonic stem cells later in life, that kind of idea. Maybe it's a way to get around some of those ethical issues. Again, maybe you can use some of the adult stem cells, get them to function like they were embryonic stem cells. If you're getting something that's not embryonic to function like embryonic, that's induced pluripotent. Pluripotent, again, can't be made into everything. They're not totipotent, but they're still quite useful. Like this, here's somebody who looks quite sick, how they drew it. And so they're getting the skin cells to kind of be reprogrammed. So they're induced pluripotent, not totipotent, but pluripotent, and then return them maybe to help the patient out. Kind of cool. So again, there's a lot of fields you can use this for. Here are some of them. There's diseases for you. I was talking about celiacs, for example, with the S&Ps. So, and I note cancer here, gene therapies. So maybe actually being able to go in and rewrite the DNA, actually start curing it. And that's actually kind of an interesting point. Last time I looked, there were 12,000 known human illnesses, not many cures. But now, in the 21st century, we're finally getting to the state in our science where we're beginning to talk about cures. So this is actually a really exciting time in human history, and we'll see where it goes from here. And maybe gene therapy is one way we can go with that. Or again, novel drugs. So we create a drug for you. Maybe not so much this like shotgun approach and hope it works on you. We actually create it based on your DNA. Or there's insulin, what I started this whole lecture on. And there's hormones we can do, vaccines we can do. I mean, there's a lot that we can do. This is not easy stuff. It takes a long time to go through research, go through trials, see if it works, look for problems. Certainly you can do trials, looks great, then put it on market, and now instead of 10,000 people having it, 10 million people have it, and problems begin to show up. So this is very difficult science, but we're doing our best. I'll also leave this one to you. Here's a goat that's living a life so that we can have what we want, milk. We're a bit of an unusual animal where a lot of humans drink milk throughout their entire life. Bear in mind, only about 60% of the human population can actually process milk as adults. This is a genetic issue based on evolution. My culture coming from the northern latitudes, most of us can process milk. I could have milk no problem as an adult. And that makes sense because evolutionarily, a very harsh environment to live in, like Northern Ireland and Northern Scotland, very hard living environment. So that gene for being able to process lactose never got turned off. It ranges like at least 90% of the population can still process milk as adults. But a lot of people still like it, even if it causes tummy issues. And again, about 60% of people out there, when they reach adulthood, can't process it. But that's still a lot of people who drink milk. So how do you produce all that much, especially because we're another unique animal who doesn't drink their own milk? So how many goats do we have to have this, and cows do we have to have this to create these factories and get them to continually make this material? And I'll leave it to you, the ethics again. Is this okay? Is that a decent life? This is only something you can answer. So, wow, yeah, this one always breaks my heart. So forensic evidence, I could never be in law enforcement. It's just not something that is for me because I know these things happen. The only thing I would say on this is, one, don't do the crime, because with today's science, you're going to get caught. And on the other side of that, if you get arrested and you didn't do the crime, get this testing done, these genetic profiles. Again, nobody should have the same DNA except you. So unless you have an identical twin, and that's another discussion, mind you. So when you're going to test amongst people, you're looking for the restriction fragment link polymorphisms, and you're looking for the short tandem repeats, these little repeats that happen again and again and again. They're specific to people. And so when outside of the identical twin idea, when a crime happens, this used to be massively expensive. Back in like the 1980s, we had the technology, but who's going to pony up the thousands and thousands of dollars? Today, not that this is Ancestry.com, but think of like Ancestry.com, a genetic test. It costs like what, $59 when they have their specials. So maybe in a crime case, pony up the couple hundred, maybe a couple thousand, whatever it is. I've never looked at the price, but get the money because you didn't do it and get tested. This poor gentleman, I can't even imagine 17 seconds being in prison for a crime he didn't commit, let alone 17 years. This poor man. And so they had semen they collected from the victim, and you can see the short tandem repeats. They tested it genetically, and you can see what these repeats were. Mr. Washington, who was innocent, the poor guy. Notice his repeats didn't match. He did not commit the crime. This gentleman, I call him that, this person did. You can see it matches identically 17, 17, 19, 19, 13, 13, 16, 16, 12, 12, 12, 12. He did the crime. And he later, of course, pled guilty. This poor man. I mean, how do you, I was gonna say sorry, but that doesn't even come close. You shouldn't spend a second in prison, let alone 17 years. So with today's technology and the pricing, get the test done. You know you didn't do it because the technology is out there now. Back in the 1980s, it might have been hard to do, but now 2022, get it done. Same with this environmental cleanup. This brings up a lot of thoughts to me. Genetic engineering can be used to clean up like oil spills, but is that always a great way to do it? It's like a tool in the toolbox kind of idea, and there are other ways you can do it as well. Fungi have been shown, and not even modified fungi, have been shown to do wonders for things like oil spills. But this is one of the other things you might do. Here's agriculture again. So now you're genetically engineering the crop to resist something, whatever it might be. So you take a gene from one thing, put it into the plant. Monsanto was famous for this. You might have heard of that. A lot of people have like a feeling towards Monsanto. I'll leave that to you. By the way, Monsanto doesn't exist anymore. It was bought out by Bayer Corporation, the same company that makes the aspirin, Bayer aspirin. So, you got to kind of keep these things in mind. But again, you're engineering, in this case, to be herbicide resistant, pest resistant. There's salinity. I brought that up earlier, so resisting against salts, that kind of idea. Increase crop nutritional value, whatever it is you're trying to work with the plant, which leads, of course, to this genetically modified organisms, which of course leads to genetically modified foods. Now, right there, right on that topic, when you look at genetically modified foods and organisms, okay, the idea is this: to increase crop yield. I think I just read an article where we formally hit 8 billion humans on the planet. Is that too many? Is that too few? That's another discussion going on. But how do you feed all of them? So that's the idea of genetically modified food, to be able to increase crop yields, hopefully increase it safely, and make this nutritious. But is it safe? Well, that's a difficult discussion. So again, this is kind of hard. Some things make sense, some things don't. Golden rice comes to mind. Golden rice, there you go. This is not like saffron or anything like that, or yellow rice. This is golden rice. You can see it here. This is rice that's genetically engineered to be heavy in beta carotene, the precursor to vitamin A. The reason why we do that is there are some places on the planet where the soils are naturally deficient. They just can't give you what you need. So we genetically engineer this, take this rice to those places on the planet, so when people eat the rice, they get what they need nutritionally. Now here in the United States, we probably don't need that. So while white rice isn't all that healthy for you, it's like eating kind of white bread. Again, it's not necessary to eat the golden rice. So this is a positive example of using a genetically modified food source. So it's one thing we can do. Here's another one you actually might have had: white wheat bread. Now, and this is a case where we genetically modified the wheat plant to be an albino. So a lot of people don't like to eat wheat bread. They prefer white bread. White bread is not healthy for you. Same with French breads and Cuban breads and all that kind of idea. So what they did was, well, let's play with it a bit. And so people think they're eating white bread, but they're not. They're actually eating wheat bread. We just, again, genetically engineered the wheat plant to be albino so that when you mill it out, it doesn't come out looking kind of reddish brown. It comes out looking white. I tried it. Yeah, it was kind of neat to try once, but I don't really eat much bread anymore anyhow, even though I miss bread. So it doesn't really come up much in my world. But it is a point now that people either like or don't like genetically modified organisms. So like here, you can now walk around Publix or Sweet Bay or wherever you want to go, or Sam's Club, I don't care where it is, and look for this, that it's a bit of a selling point, saying no genetically modified organism was used in this food item, and leave it to you to make the choices you wish to make. This is different than organic foods, mind you. So again, kind of keep the topic separate. And there you go. You've made it to the end of this chapter. Again, if you have any questions, feel free to let me know.